So welcome to Demography Today lecture series, sponsored by the BBVA Foundation and co-organized by the LOMPOP Horizon 2020 project and the Spanish National Research Council. And we have the pleasure today to have here Alberto Palani. Alberto holds a PhD from the University of Washington, Department of Sociology, and currently he's a researcher at the Institute of Economy, Geography, and Demography of the Spanish National Research Council, where he's the principal investigator of the ERC Advanced Grant ECHO project. And he's Samuel H. Preston Emeritus Professor of Population at the University of Wisconsin-Madison, and as well at RAND Corporation. Previously, he has been a professor of sociology and demography in several US universities. He served on the PAA board of directors from 1996 to 1999. He was vice president in 2004 and president in 2006. In 2019, he has been awarded the USSP Laureate Award, and he is as well Doctor Honoris Causa for the University of Leuven. He is known for his research contribution in the areas of health, morbidity, and mortality, indirect techniques for demographic estimation, demographic models for HIV, AIDS, aging in developing countries in general and Latin American in particular, fertility and family demography, and more recently the relationship between early childhood health and adult health and mortality and social stratification. So welcome, Alberto, to our lecture series. As usual, we have between 45 minutes to 1 hour talk, and then we open the floor for questions. So
I just wanted to use the title to tell you a little bit about what this is all about. But this quote from Haldane, I think summarizes very well what I would like to do. So the idea here is that for a scientific theory to have any value, it has to have predictive value. And what I'm going to propose to do is to generate conjectures based on quite solid theories that could be tested, and then to use them to predict. And what we're going to predict is obesity, type 2 diabetes, and other comorbidities that are very common in low and high income countries. So I will start by asking a few questions. As you probably know, the incidence of type 1 diabetes has been increasing, and the age of the onset of diabetes, which used to be fairly low, is also increasing. Why is it that Finnish origin children who are endowed with similar genetic heritage, but they live on two sides of the border, Finland and Russia, those who live in Russia have no diabetes 1 at all, no signs of it, whereas those in Finland have one of the highest rates of diabetes in the world? There's another question that I pose. Why is the incidence and prevalence of obesity and type 2 diabetes has been increasing so rapidly everywhere, but the speed of the increase is usually, as I will show you in a second, much faster in low to middle income countries? Finally, populations that have equivalent levels of child malnutrition, stunting, but have dissimilar adult diets, some modern, some more traditional, they experience completely different obesity and type 2 diabetes patterns. One much like the US and Mexico, and the other much like countries that experience almost no prevalence of those conditions. So these are questions that I'm just putting there because part of the problem that I try to solve is this. This is just evidence of what I just said. On the left, you have childhood diabetes in Norway. I couldn't get the data for Finland. And you can see that there is an increase. Now, why would type 1 diabetes in children in Norway be increasing? We know that those countries have very high prevalence of diabetes 1, but it's unclear why it's increasing. And the other one is showing you the relationship, apparently, between diabetes 1 in several states in the United States and exposures to chemical. And on the left-hand side axis here, you have production of some chemicals And on the right-hand side, you have the rates of incidence of diabetes type 1. And you can see that there is a clear relation. I'm not establishing a causal relationship here, but only suggesting that something amiss is there. This is obesity rates in countries of OECD. And you can see that the speed at which they are growing are pretty similar. And the most outstanding here, Mexico and the US are on the top, but other countries appear to have been following the same trends. This is a more convincing portrayal of the extent of obesity. There are two things that I would like to point out. The first one is that if you look at those who are still quite low here, these are all countries with one exception, Japan, that have not completed their quote-unquote mortality transition. They still have relatively low levels of life expectancy, whereas all those who are here have completed the transition and they have gone to life expectancies that exceed 60, 65. Notice that there is a mixture of countries, very developed, United States, very undeveloped, like Egypt, or a mixture like Argentina, Mexico, and Canada. There's not very much of a predictive variable to use income or GDP to predict obesity. Notice that the levels of diabetes are reaching on this side about 45%, and that there is a difference between males and females. The difference between males and females is going to be important for something that I have to say later. This is just to show you a more detailed picture of the prevalence of obesity. And by the way, obesity here is overweight plus the obese, 30 and above. And I just wanted to point out this. The prediction is that by 2030, the percentage of people who are going to be obese is about 50%, half the population in the US. And it's likely that if you do a projection similar to this, in other countries you will find similar quantities. This is a map. The red part of the map is the high prevalence of obesity, and you can see that it's just about everywhere. Nordic countries with high levels of development, middle-income countries like Mexico, middle-income countries like Argentina, Chile, and then you have poorer countries in Northern Africa that also are experiencing the same phenomena. This part over here is just the Arabian Peninsula. But what I'm trying to say is that the curse of obesity appears to be quite well distributed independently of income and geography. This is the distribution of countries by prevalence of diabetes. If you do a little exercise with your imagination, probably 40 years ago, this distribution was shifted to the left. And they were all concentrated around between 0 and 20. In about 50 years, this distribution shifted to the right, and it is what it's now. Worst part is that children are experiencing the same kind of phenomena. Children are also becoming more obese. And the ranking order of countries of child obesity is more or less the same as the ranking of obesity among adults. Here you see less of a sex difference, except in some countries like South Africa, where girls appear to be a lot more obese than males. This has importance because there is a correlation between maternal obesity and child obesity. Another point to draw from here is that the best predictor of obesity as an adult is obesity as a child. So from here, I could actually, the correlation is around, 0.6, 0.7, and barring extraordinary circumstances, I could predict what the adult obesity 20 years from now will be in Portugal, just by looking at these numbers here. This is a curve of obesity, childhood obesity in the US, drawn from the National Health and National Health Survey in the US, and it shows the steady increase. There's some noise here, probably produced by the entrance of different cohorts, and that does appear to be that those who are younger are dropping back in the rates of obesity. Finally, accompanying obesity is type 2 diabetes. This is a map of the prevalence of type 2 diabetes and 1. They are combined, but the prevalence of diabetes 2 overwhelms diabetes 1. And notice where the dark areas are. Here, this is Northern Africa and the Arabian Peninsula, and then Mexico again. And you could go back and do a prediction in the future, which is what, oh, excuse me, I wanted to show you also in Latin America. It turns out that countries like Brazil and Ecuador and others that are even poorer, like El Salvador and Guatemala, are experiencing mortality rates of diabetes that are growing very, very fast. As I was mentioning before, you can use the obesity data to predict diabetes in the future. This is a worldwide projection done by the World Health Organization. They are forecasting that by the year 2025, there will be an increase of 72% in people with diabetes 2. But it's so unequally distributed that that number means almost nothing. Look in Africa, they're expecting a doubling of type 2 diabetes. And in the poorer areas are the worst affected. Then you have Southeast Asia, among other things. And then there, again, the Arabian Peninsula and Northern Africa. Altogether, if you convert this number into a prevalence worldwide, it's about 10 to 15% expected of people with diabetes type 2. Okay, 3 problems. How do we explain this? The emergence of an obesity, quote-unquote, epidemics and associated type 2 diabetes. And mind you, the type 2 diabetes is just one disease that goes associated with obesity. There are others. And there are others that are also associated with type 2 diabetes. So we're looking at the tip of the iceberg here. The second question is, what are the predicted impacts on comorbidity, disability, and mortality of these trends? And the third problem, which is the one that I'm interested in, is what does this mean for the evolution of human health and longevity in the long run? So those are the 3 questions that I'll try to answer, or propose a way to answer it, because I don't have an answer. So this is the research agenda of the so-called ECO project. And I want to mention 3 things about the ECO project that provide a scaffold to answer the questions that I just asked. First, there is theory. And the first thing is that the project is based on 3 pillars. A pillar that is developmental biology, another is evolutionary biology, and a third one that is not hidden here is demography, which is what I know how to do. I don't know how to do the other 2 things. There is a new movement in biology called by the acronym ECO-DIVO-EVO, which joins together evolutionary biology, ecology, and developmental biology. It is within this framework that I would like to try to develop conjectures to explain the phenomena that I just mentioned. Within this, there is what is called the developmental origin of health and disease theories of biological development, which it's known broadly as DOHA, and I will use the acronym throughout. The consequence of having DOHA as an umbrella to produce explanations for what the 2 phenomena that I mentioned is to understand the role of epigenetics. And the third element that comes here is to understand the role of the microbiome and in general the role of symbionts and holobionts. So that's the part of theory. The part of models is that one needs to model first molecular processes that are determining individual propensities for health risk, in this case diabetes and obesity. Second, they should be joined by life cycle models with overlapping generations, which we social scientists do very well. And the third thing is to model randomness explicitly, not as a nuisance. Most social scientists try to get rid of randomness in a variety of ways, and you correct for standard errors that are affected by clustering, but you don't worry about the effect of clustering, which is what one should be modeling. It's an example. The third part of this is that we need data that are new. We need data like we normally collect, integrated standard demographic and life course data, but with enhanced biomarkers. And here I would like to emphasize the need for using multiple tissue specimens for epigenetic and microbiome analysis. The stuff that is being done now down with the SNPs and GWAS is okay, but is not sufficient. I think utilizing epigenetic clocks and microbiota genome is a much better strategy. The final thing is to implement multi-cohort longitudinal studies collecting, as always, but better, the socioeconomic data and demographic and health information that was mentioned before. In theory, if one could do joint human-animal studies, it's even better. This has been done already with the microbiome, where the researchers have injected microbiome material from humans with some conditions into mice, in order to understand whether the effects of the microbiome of a human affects the development of conditions in the mice, and the answer is yes, it does. So if one could do that, follow that kind of model, it would be perhaps the best strategy of all. This is the outline of the rest of the presentation. I will first give you a little background of the brief history of human mortality, which we all know, but it's so common that we get used to the details and we don't realize how impressive an achievement this has been. So that's the first thing I will do. I'll try to go as fast as I can. And the brief history of mortality decline is characterized by being fast and compressed. This is a very famous graph produced by Fogel, and his idea here was to compare over the, let's say, 11,000 years of history of Homo sapiens, the speed at which certain things occur. In this case, this part is the population growth, and along this, he plotted all the various very important events. Most of the scientific events of interest occur here. And it is there where you see the population emerge after 10,000, well actually 200,000 years of being almost zero, it grows and shoots up like this. So did the amount of energy that we produce. More importantly, if you overlay that with life expectancy, which is the measure that I will use throughout, you can see that the growth of life expectancy is almost as sudden as the growth of the population and the growth of energy production. And you lay obesity, and it's even worse. The growth of obesity is even faster than the growth of life expectancy. This is important for what I will have to say a little bit later. Now, this is just to entertain myself, I think. How fast is this? Well, if you look at the history of Homo sapiens from, let's say, 250 to 300,000 years ago, the life expectancy increased from about 25 years to 80, but only in the last 200 years. So do this exercise. Compress the history of Homo sapiens to a year. It means that the decline of mortality occurred in the last 7 hours. That's pretty amazing. It means that also in the past, we were able to gain 1 year of life expectancy every 20,000 years, and now we're gaining 3 years of life expectancy, 1 year of life expectancy every 3 years. This comparison is even more important for what I would like to say later. There's a massive disharmony between two things that matter, the change in the environment and the change in the genome. First of all, think of doubling times now. The doubling time for the life expectancy in Europe was 119. It took 119 years for the life expectancy to move, let's say, from from 30 to 60. In developing countries, it only took 67 years. The doubling time of obesity is only 32 years. But now, suppose that I want to calculate, and this is kind of pushing the boundaries of what is actually legitimate to do, the doubling time of the genome. Well, if you use as a point of departure the rate of mutation, which is over there, calculated according to Elwin Scully, I get a doubling time of the genome of 138 million years, meaning that we as species have moved the environment, and by environment I mean include mortality and the life expectancy, much, much faster than our genome has been able to change. It's also, the history of mortality is also very highly heterogeneous. It took Cuba and Japan about 1/3 as much time to get the same life expectancy. This is a graph of Sweden life expectancy for males, for females. This is a graph for Cuba, the blue bars, and the pink bars for Japan. I forgot to put here the progression of life expectancy of countries in Northern Africa. If I had, you would have had a graph that begins here, extends here, and then all of a sudden it shuts up with much more speed. Why did this happen? And I think the only culprit, if you go back to the graph of Fogel, is that we generated scientific knowledge. An example is germ theory, that produced in technological improvements, let's say antibiotics or vaccine, and human organization that spread the changes induced by germ theory and antibiotics. That's the main sort of force of all this. That led to very important nutrition improvements in terms of number of calories and quality of nutrients, and then a massive host pathogen balance shift that made the attainment of reproductive ages an easy task, once a task that was very difficult 2,000, 3,000 years ago. So what about the future of longevity? Will these improvements in survival that we've experienced so far persist? And to what an extent does the answer depend on the influence of early conditions? So I'm now zeroing in on the problem that I started with. Two sets of predictions. There are two sets of people in this field. Some of them think that human lifespan has no intrinsic limit, and some of them think that human and any mammal lifespan has hard limits, and it can only increase up to that hard limit. You probably know this figure. This is a figure that was drawn by Jacques Vallin and Franz Meslet, following some work by Vaupel and Opel. And essentially what they try to say is, look, if you go back to 1840 and you map life expectancy, it grows into a line, and you can project this line into the future as evidence that probably longevity will continue to increase in the same way. Projections don't say that. The UN projections say that life expectancy will depart from that trend. So what is the prediction of these people? That probably this is what the trajectory will be in the future. There are some other people, and this is based on an article that appeared in Nature a couple of years ago, that sustain just the opposite. These are just 3 pieces of evidence suggesting that there is a hard limit on the life expectancy of humans. I won't go into the detail. I will only tell you what the conclusion was. The conclusion was that by analyzing global demographic data, we show that improvements in survival with age tend to decline after age 100, and that the age of death of the world's oldest person has not increased since 1990. Our results suggest that maximum lifespan of human is fixed and subject to constraints. So we have those 2 kinds of positions. My problem is somewhat different. I don't care whether or not the lifespan is finite or infinite. I care about what the future trajectory is going to be. if it is infinite, how do we approach this infinite boundary? And if it's finite, how will we approach it? Would we have to go back? We would have decreases in life expectancy? We would oscillate around some value? And to answer that, one needs to take into account, into account the influence of new behaviors, diet, physical activity, consumption of substance, etc., external threats, which are very much in the newspapers today, including global warming and the resulting reemergence of new diseases. But for me, the most important thing is what will happen with the cohorts that were born, say, after 1950, and that would be born in the next 20, 30 years, which will compose the bulk of the aging population after the year 2030. And the claim is that it's likely that because of the composition of those cohorts, that the increases in life expectancy will stop. We may have a backward movement, or we will have a long period of a stalemate for a time. So let me peel the onion now. I will go back to the outline, and I want to focus on Doha. And Doha has a framework, as an umbrella to permit an explanation. Doha refers to a variety of theories that have been evolving over time, but that are based on a very simple principle, and that is the principle of phenotypical plasticity as an evolutionary adaptation. In evolutionary adaptation that has been successful, and it consists of the following: that phenotypes may change at the prompting of some environmental cues through some mechanisms that we will like to describe, but the key point here is that genotype is not changing. That is, the phenotype changes despite the fixity of the sequence of base pairs. These two guys here are two little creatures. The insect world is full of examples of phenotypical plasticity. I don't know if you've known about, this is the so-called stick-back fish that lives in oceans, but especially in lakes in the United States and Canada. And it's a fish that, when sensing predators, the offspring of certain mothers will develop these little sticks that are there that are like an armor against the predator. When they sense that the predator is absent, the offspring don't have it. So they change the phenotype. They are, genotypically identical, but they've changed the phenotype in response to the environment. This is a locust from the African savanna, which can be in two kinds of conditions. It can be gregarious and have very long wings and be very hungry, or it can be very solitary, very short wings, and with a very austere diet. The change from one to another depends on sensing crowding. And to the extent that they sense crowding, the offspring of the next generation will have one type rather than the other, the maternal type. So these are examples in the animal world, but the Doha, this theory that I was referring to before, is using it to explain certain outcomes in mammals and humans. So a little kind of tour on what phenotypical plasticity is. There are two types, the reaction norm and the polyphenic. Reaction norm are changes in the phenotype that are gradual in response to gradual changes in the environment. The typical example is muscle. If you do exercise with weights, you'll get more muscles. The more weight you use, the more muscle you get. The polyphenic is a discrete change in the phenotype given an exposure to a cue, and that is the example of the stitchback fish that I show you on the locust. The important thing here about phenotypical plasticity are what the mechanism behind it. And the most important is a direct mechanism where there is a modification in the proteins produced by the series of genes that the individual has. These modifications occur during transcription, translation, or can be during post-translational regulation. And it's all epigenetics. In addition, there is the role of what they call small non-coding DNAs and the small interfering RNA that alter proteins. There's another mechanism, there's neuroendocrine that involves in the hypothalamic axis, and it's just the production of hormones that may lead to inflammation or protein synthesis. Both can change the phenotype. And finally, there is signal transductions that is mostly a mechanism of communications between cells. So those are the three mechanisms through which phenotypical plasticity works. Key thing here is, when does phenotypical plasticity develop? The cue has to be early, and it can occur anywhere in gametogenesis when the gametes are being produced, during early embryo development, very early, even before implantation, and during the midst of the embryonic development, gastrulation, neurulation, and then later when the organs began to form. Also, there is the possibility that changes can occur neonatally. The duration of the response, it can be, if you remove the cue, maybe the changes do not occur, or they go back to the original. That they can be durable, and that means that they can be passed on from one cell to another during mitosis, or, and this is the most doubtful part, it can be transgenerational and transmitted from one generation to another. I wanted to mention one final thing here. In phenotypical plasticity, usually, not usually, but there are many examples where the cue is actually transmitted by symbionts, that is by, let's say, microbes that live in the organism in question. And it is that microbe and the production of metabolite of that microbe that actually leads to the reaction. It's the case of the microbiome among humans. I want to say something about the various ways in which early condition can affect, can have delayed effect. There are at least 5 classes of pathway. One is maternal constraints. That is, this is the simplest form. Small mothers give birth to small children, and it has to do with physical constraints that actually limit the size of the birth. This is just, it's studied a lot, but it's not of interest here. The second pathway is via infections. This is individuals who are exposed to certain infections during childhood may develop later some chronic disease. The better known examples are the Helicobacter pylori, which causes stomach cancer, the rheumatic heart fever, which causes mitral valve stenosis, and then the flu, certain strains of the flu that may actually alter adult immune function. Just to give you, think about this. Rheumatic heart fever is very prevalent in developing countries up until 1960. And those individuals who were saved from rheumatic heart fever because of the use of antibiotics, but who contracted the disease, will survive to be age 60 or 70, which is when the problems would develop. So you should see an increase, everything else constant, in that kind of disease when those people reach the age of 60. And that's simply because the individuals who experience rheumatic fever, which used to die before the age of antibiotics, don't do so anymore. This is an important thing that we'll come back to discuss. The other pathway is the existence of recurrent infections that induce inflammation. This includes chlamydia pneumonia, the contraction of ailments and periodontitis that lead also to inflammation, heart disease, and hypertension. Then comes the so-called Barker hypothesis, another which is a different mechanism, and this is a mechanism that operates by constraining the growth of organs. After there is a sensing, this is the cue, that nutritional conditions are bad, the fetus will develop mechanisms such that it will impose constraint in the growth of the number of cells, for example, that go to produce nephrons. And so in favor of, say, developing the brain or the heart or the lungs, which means that this individual, if born and survived past age 5, will be at risk of having hypertension if there's a lack of nephrons in the kidney. This thing has been called the thrifty phenotype by Barker and Hale, and it was thought to be an evolutionary developed mechanism to trade off between fitness now and fitness later. That is, in natural selection terms, you would prefer somebody to survive, let's say, at least to age of reproduction than to survive beyond that, because beyond that, from the point of view of fitness, it means nothing. Therefore, the trade-off here is develop the organs that will make this individual get to age of reproductive function, and do not develop those organs that may give him a disease after the age of reproduction. A few years after Barker, there were two sets of extensions of this theory, which led to the formulation of what is called now PAR, or the predictive adaptive response, which involves stresses that are not just nutritional stresses, but involve social stress as well, and nutritional stresses that are not just a contrast between very acute and not very acute, but rather a gradation of nutritional stresses, both producing the same kind of reaction. That is, if the individual being developed senses, say, abnormal conditions, it will guide the development plastically to accommodate to that development. But if they find conditions that are inconsistent with those that have been sensed, then some conditions may develop later on. This is what is called the phenotypical plastic basis for the Doha theory. The point is that most of those phenotypic responses depend on modification of the way the DNA works, which positions itself, the chromatin. There are two variants of this theorem. Probably they are not important, the distinction, but I'll mention them. Some of them suggest that their response to cues is not short-term. That is, a short-term response would be, for example, a fetus senses malnutrition today, and it reacts by producing accommodations to be born 9 months later. That's a short term. The longer term is when the response is not to the conditions today, but the conditions several generations ago. And the idea here is that fetal development may respond to conditions that were experienced by grandparents, not just the mother. That's a different kind of theory, but sort of the distinction is not very important, at least in this presentation. In all cases, phenotypic plasticity is thought to be an evolutionary adaptation that improves average fitness before reproduction. But the problem is that it may backfire. I don't know if you've seen examples of animals who, depending on the season, change their coat, say rabbits or foxes, or those who live in the snow, for example, in the winter, they change their coat to be white, so they're less identified by predators. But if there is no snow and they continue to produce the white coat, they're going to be in trouble. This is quite prevalent. Something similar appears to be happening with obesity and type 2 diabetes. that just as for mammals in the wild affected by global warming, obesity and type 2 diabetes may be partially as a result of mismatches between the environment that was predicted when the embryo was developing and the environment that the child who was born encounters, either immediately after birth or soon after birth within the first 5 years. It could be. So this is where I'm trying to go. And I want to just make a few points here about obesity, which apply to type 2 diabetes as well. First of all, there is a correlation between maternal and paternal and child obesity and type 2 diabetes. 2, people who have excess body weight tend to marry people who have excess body weight. The consequence will be that there will be less variability, and therefore the offspring may inherit more likely the obese type of the parents. The third thing is that there are fertility differentials between people of different body masses. And then the fourth thing is that there are disparities across population by social class and ethnic groups. So if obesity and type 2 diabetes are in fact the effect of mismatches, and all these 4 conditions apply, they will be very important to predict obesity and type 2 diabetes in the future and to understand social class and ethnic group differentials. So the question that people ask is, how can this trait, phenotypic plasticity, which is supposed to be so great, how could it have survived the natural selection? How is it that it's still with us? And the answer is that it's a phenomenally successful adaptation that increased fitness when facing rapidly changing environment, or even in cyclically with long cycles. And the machinery of this adaptation that circumvent the DNA circuitry and produce plastic responses that are more sensitive to rapid environmental changes and phenotypes, more adapted to them. Some of them are heritable. And finally, there are trade-offs to attain reproductive ages and maintain fitness. The problem with us is that we invented antibiotics. We discovered what bugs were, what parasites, how parasites could control. We extended our life expectancy beyond age 40 or 45, and now we're facing the results of trade-offs that we're operating on the assumption that we will only live until we're about age 45. Let me, I would like to emphasize this part, this mechanism of phenotypical plasticity, the one that operates by direct changes in the structure of the DNA, not in the DNA per se, but in the structure, so that there is there are impacts on the probability of transcribing, translating, or post-translating the product of a gene. So I will go to this. So this is this third thing. How is the time going, by the way? Oh, what does that mean? Huh? 45 minutes already? Well, I'll go 15 minutes at most. So what is epigenetics? Mario is an expert here, so I'm going to go very fast so that he doesn't catch my lack of knowledge of this. But this is the main mechanism through which some of the phenotypical plasticity we're interested in operates. There's two quotes that I find very nice that tell you exactly what epigenetics is all about. The first one is by Castleman. It's a genome that you were born with is not the same genome you die with. And understanding by genome not just a set of base pairs that you were born with. And the second one is by Ziff, an evolutionary biologist, who says that epigenetics confer individuals with the same DNA, different phenotypes that are adaptive and plastic and tuned to the phenotype to environmental cues. It can be very good, but it can also be very bad. And epigenetics have been linked to all sorts of diseases. This is a picture of them. There is one that is missing, dementia, and has been recently related to it. The guy who is here in the middle is a fetus. Actually, epigenetic changes may occur way before a fetus is completely developed, in the very first stages of the embryo formation. But they can also occur later. So I don't know where to go through this. I only have 15 minutes. Just think about this. You have a bunch of cells in different tissues. They are all, quote unquote, identical. They have the same DNA in the nucleus, and yet they do different things. You don't grow eyeballs in your head. Your eyes don't produce insulin. So how come? How can that be? The other thing that is interesting is there are some genes that are not expressed unless they come from a particular parent. These are the so-called imprinted genes. How can that happen? And the final thing that is, being a very naive student of genetics blew me off was that women are kind of mosaics, because having two X chromosomes, one of which needs to be eliminated to reduce the excess load of base pairs, the deletion occurs randomly, and so different cells have a different chromosome that has. Now, there's another graph that I had here about twins' resemblance in longevity and schizophrenia. It turns out that the resemblance measured by concordance coefficient is no more than 30 or 40%. How can two identical twins differ in that risk of getting schizophrenia if they are identical genetically? Well, is it randomness? Yes, there's a lot of randomness in this. Is it environment? That also. And is it genes? It's part of the explanation. It's a bit of everything. This is a graph that shows the development of an individual from embryo and how it's subjected to the influences of genetic and epigenetic inheritance, and the effects that move over the life cycle involve developmental stochasticity, that is the role of chance, and environmental influences. Mario did a very nice study some years ago showing that twins' epigenome differ as they grew older, suggesting that there's a lot in the epigenome that is actually modulated and shaped by environments. What is the mechanism through which epigenetic works? I'll just be very, very, very brief. There are a number of things, some of which are not really epigenetic. This is something that occurs outside the nucleus. But the most important thing are the role of methyl groups and acetyl groups that are essentially molecules that attach to the DNA, to some of the base pairs or to some of the histones, and actually relax or contract the DNA. If you contract the DNA, then all the machinery for transcribing can't find the proper location of enhancers or promoters, and the gene remains silent. But if you relax the DNA, that can happen. Acetyl groups mostly relax, methyl group mostly repress. And epigenetic is just a big orchestra that works around these two musicians, as I call them, and they produce very different symphonies. Then there's a few other ways in which they call epigenetic when the messenger RNA comes out of the nucleus. It may or may not end up producing the protein. There are some RNAs that may impede that. And then there are still other molecules that can misfold the proteins and they become useless. Anyway, so these are all mechanisms through which epigenetic works. There's a fantastic book, it's called Extended Heredity, that goes over some of this. And there's a lot of questionable things there, but suggestive. Okay, the question is where do they come from, this methyl and acetyl groups? And I think that the full story is not yet known, but we know that there are substances that are methyl or acetyl donors, and some nutrients are important. And this is something that relates malnutrition early on in life with development of disease later on. One of the most important is folate, or folic acid. And the key thing in my talk here is that some of these are synthesized, are produced by bacteria that you have in your stomach. So what you eat or what your mother ate when she was pregnant may have altered the number, the percentages of methyl or acetyl groups that you had available for either silencing or expressing your genes. When does this happen? When do methyl and acetyl groups sort of become active? For what I am interested in is during early in life, even in the formation of the gametes, in embryogenesis, neonatal, these are all crucial periods. But there are also other periods in life when you could actually modify epigenetic marks. These are just examples of how methylation or acetylation has actually affected the function of some organs. For example, there's a very famous guy who works on stress, this guy Meany, who's done a lot of work with rats who are subject to grooming, that if you stop grooming, there is a methylation that leads to rats that become very aggressive. And this is an example of the study of the Dutch hunger survival, where they also find some methylation anomalies in people who were affected by the... Anyway, so these are all... this is a graph that shows the probably development of high-risk obesity and high-risk diabetes via epigenetic marks. I'm going to skip a few of those things. I want to get to the issue of let's see. If you want to know about the microbiome, I can send you the slides. The microbiome is, for me, is one of the most important quote-unquote organs, because it is an organ that actually produces substances that become methyl and acetyl groups. And they are fundamental in the development of children, and they have been linked with a lot of diseases. I'm just listing a few of them. And the way in which it works is as follows. The microbiome that you're born with is entirely due to your mother. You're usually sterile up until you come out of the womb, although they've been discovering bacteria in the placenta recently. So that's a little bit up in the air. Most of the bacteria are donated by the mother, and largely they are set by age 3 to 5. But then during adulthood, you can modify this by infections, use of antibiotics, by your diet, by exercise, by chronic diseases. And this is the experiment I was referring to before that shows that, in fact, the microbiome contains microbes that can make you obese, that can lead to development of diabetes 2 and other ailments. This is what I mentioned before, that is that the microbiome is implicated in methyl and acetyl production. And so this is a summary. I promise I won't exceed the hour. So now the question is, do this mechanism always operate? And in particular, can the delay effect of adverse early conditions be blocked later in life? Can you intervene and change something that was set when you were in embryo? Are there critical ages for the delayed effect? Can they be altered? Can the delayed effects be triggered earlier than critical ages or postponed beyond that by exogenous shocks or intervention, by stress, by nutritional deprivation? And if damage accumulation is required, what are the thresholds? And this is the kind of question that I would like to sort of answer by using the data that is available, by using new data, but particularly by using formal models. And I suggest two types. One, which is a simple one. It's the only one I will talk to you about, that consists of the following. I want to know whether early conditions really matter or not. And what I need to do is to actually project cohorts of individuals that vary in terms of the propensity to be born with conditions that make them more at risk of developing adult conditions. Irrespective of what is the epigenetic process that led to those predispositions. The project that I'm involved with is trying to investigate those processes, and in addition to do what I'm going to do right now. But right now I don't have any information about the processes themselves, and so I'm going to just talk to you about a very simple way of assessing whether or not early conditions really make a difference in adult mortality, in this case, or adult disability. So here's the idea of them all. So you have the DOHA predicting predictive adaptive responses. You have disease early in life and related to adult conditions. You may have sustained inflammation. In any case, individuals are born with a propensity to develop adult conditions, and that will modify the phenotype. And this will occur only after long periods of latency. Beyond some critical age, say beyond age 60, you will actually express those early conditions. And these are the questions that I want to ask. The model is as follows. I imagine a simple death process. There's those two states, you're alive or you're dead, for multiple birth cohort. And I only focus on the force of mortality. Then I introduce, quote unquote, the fetal programming part that essentially says individual members of a cohort have different probabilities of developing delayed effect because they are born with early conditions that are different. And the model is very simple because the only thing that it does is to say that individuals are born with a particular set of propensities, not where those propensities come from, which is what is of interest to us. The intuitions behind the model are this, and I'll just summarize them in a sentence. Imagine you are in a society like, I don't know, Uganda, where most kids before 1950 died of infectious diseases before age 5. The mortality rate at that time probably was of the order of 300 per thousand. So you had a third of the birth gone before age 5. Suppose that now you come in and you distribute, as they did, antibiotics that knocks out a number of diseases that kill these kids. And now instead of having a mortality of 300 per thousand, you have 50 per thousand. So now you have a set of individuals born in a cohort or in several cohorts that will survive probably to age 60, which are likely to have been damaged by adverse conditions, nutritional conditions early on in life. That increase in survival that we concocted by inventing antibiotics that led to the decline in mortality that I showed you before may be the cause, the trigger of an increase in obesity and type 2 diabetes past the age of reproduction in this case. So if that's the way things work, then I should be able to actually simulate what would that do to the mortality of those people who reach age 60 and essentially forecast mortality in the future. If somebody was 50, he will be 60 in 2010 and older later. So I could, in theory, predict the mortality of those people if I know what fraction of them is exposed, has higher propensity. These are the main parameters of the model. I just want to show you the results and I will stop. This is just to show you that I know some calculus, but the importance of these two formulas is that the prediction of the model goes against every single theory of mortality that you've read about. And in particular, there's a number of things here that my colleagues think are somewhat controversial because they go against a lot of the practices that demographers have been using in the last 50 years or so. Essentially, the most important one is that that mortality at older ages increases at a constant rate, the log of the mortality. It's called the Gompertz Curve. It has always been thought that it fluctuates within a very narrow range and that it doesn't change very much. Well, this model suggests that the slope of mortality may change as a result of the influx of these people who have higher propensity to develop adult disease due to adverse early conditions. I just want to show you a figure and then I'll quit. We've done microsimulation, we work with real data, we work with invented data, and the results have always been the same. So let me just, so this is a comparison. I'll go to the worst country that we use. We use Guatemala, which is not Uganda, but whose conditions with malnutrition and high levels of mortality resemble more or less Uganda. And what we did was to try to predict what would life expectancy at age 60 is, it would be, past years 2040, with or without the presence of these mechanisms associated with Doha. And the results were the following. With mild regime of delayed effects, you would get this curve. That is, for example, in 2040, life expectancy at age 60 would be about 19 years. But if you put mild conditions, that is, if the individuals who were born under precarious conditions, nutritional conditions, who survived to later ages, have relatively modest excess mortality, you would get a life expectancy. I'm sorry, I'm shifting the curves. These are the guys. These are the worst. So this would have a life expectancy of around 18.5 or something like that, which is a loss of about 3-point-something years with respect to the best one. So the question is 3.5 years important, big, to worry about? And so in order to answer that question, I will show you what other things are. Look at this. The losses due to smoking are estimated to be 4 to 5 years. So the losses that I calculated before are between 1/3 and 2/3 of those losses. And we do think that the losses due to smoking are big. So there's no reason to think that these are very small. The other thing that is of interest here is that the United Nations produced a forecast for Guatemala of the life expectancy at age 60. Between 2010 and 2050, they projected a gain of 5 years. But if you take into account what we just calculated, we would lose 1/3 of those projected gains just only because there's an influx of people who've had have experienced this early conditions problems. The project is meant to go beyond this and to actually illuminate how these early conditions start. How is it that I can predict the probability of a birth to develop certain conditions, so that to illustrate, to enrich the model, and then link that to a model of the life cycle. And this is an extension of what we did, and this is the fantasy. I'll just show you in a graph. So the idea here is to have biologists to help generate a model that will predict the probability, for example, that an embryo will develop certain epigenetic marks that would prevent normal functioning, say, of the pancreas, to link that to models about early life of that individual and how those conditions affect that predisposition, and then to link that with models of adult life course, whether the individual exercises, has a good diet, a bad diet, and how does that modify the previous conditions to finally predict what would be the outcome of interest, the probability of diabetes or the probability of type 2 diabetes, of obesity and diabetes, and the other comorbidities. This triangle here refers to ABM, which is an acronym for agent-based models, which is the kind of models I think we could implement to study, to stitch together all these 3 or 4 modules.